Hi, my name is Anagha Nair. I'm from Mumbai, India, and I'm 17 years old. My mentor is Erin, a PhD candidate at UC Berkeley. So, my research topic is what role do infiltrating T cells play in the development of gliomas? So, this topic focuses on a certain important factor in our immune system and how it impacts the development of gliomas. So, a glioma is a broad term used for a variety of brain tumors that arise from the glial cells, which is a specific type of cell found in the nervous system that provides nutrition and maintenance of nerve cells. So, some examples of common gliomas are astrocytomas, ependymomas, and glioblastoma multiformes. So there is something around every tumor which is known as a tumor microenvironment, and this consists of several components such as various cells like dendritic cells, neutrophils, tumor-associated macrophages, and of course T cells. So recent evidence has suggested that the components of the tumor microenvironment play a role in the development and the growth of tumors. And so I decided to focus on T cells, which is a type of immune cell, and how it impacts gliomas. So, to understand T cells better, you can easily compare your immune system to a law enforcement system or a country's defense system. So, it'll obviously have many subparts and units to it, such as patrolling street police officers, which is analogous to neutrophils and macrophages. Then there's military intelligence, that's analogous to B cells. In a similar manner, the immune system has T cells which act as soldiers or police commanders that will identify who the criminal or pathogen is and works towards destroying it. So there are different subtypes of T cells, and my research paper focuses on three main types, which are cytotoxic, helper, and regulatory T cells. So what is their function in the immune system? So on the surface of a cytotoxic T cell, the CD8 plus co-receptor is found. This co-receptor allows cytotoxic T cells to interact with proteins on the surface of other cells, essentially building a bridge between the two. Through these interactions, cytotoxic T cells can identify normal cells that have been infected with a pathogen. So once it's identified, the T cells are activated and they work towards killing the affected cell by releasing certain cytotoxic molecules such as granzymes. A helper T cell has a CD4 plus co-receptor on its cell surface. This co-receptor allows helper T cells to identify a pathogen peptide displayed by an antigen-presenting cell, which are also known as APCs. So APCs are a type of specialized immune cell that present peptides to help cytotoxic and helper T cells identify them. Once a pathogen peptide is identified, the CD4 plus T cells are activated and they signal other immune cells to attack this pathogen through the release of cytokines such as TNF alpha. So this CD4 plus co-receptor can also be found on regulatory T cells, which are also known as Treg cells. However, unlike helper cells, Treg cells are used for the termination of immune responses that are no longer required. So Treg cells are capable of suppressing immune responses by firstly releasing anti-inflammatory cytokines, secondly by producing molecules that eradicate activated immune cells, and finally by changing the behavior of other immune cells itself. So my research paper is a literature review, and so the methodology I followed was an extensive amount of reading of many research papers that looked at similar topics like the effect of T cells on glioblastoma patient survival rate and how regulatory T cells promote glioma stemness. I also spent quite a lot of time with my mentor trying to understand the commonly used lab methods used in immunology research so that I could better understand the research papers and its results. Finally, I condensed, synthesized, and analyzed all the research I'd collected into one comprehensive literature review that could cover as much information on the role of T cells in the development of gliomas. I also looked at and gave my inputs into future perspectives and possible therapeutic methods of T cells. So through my research, I was able to learn a lot about how the various subtypes of T cells have an important role to play in the development of gliomas. I'll now go over some key findings from my literature review. So a higher infiltration of specific effector T cells such as the helper T cell and the cytotoxic T cells can lead to an improved patient prognosis as these cells work towards initiating anti-tumor responses. Secondly, it's been established that most T cells in the tumor microenvironment are exhausted. This causes the evasion of cancer immunity. And there is also a high variance in the exhaustion signatures in gliomas, and this contributes to the lack of anti-tumor responses. As I mentioned before, all the T cells are mostly exhausted. This aids in the rapid progression of a glioma. Gliomas also have the advantage of further development through glioma stem cells, supported by the tumor growth factor beta. And this release is facilitated by an increase of infiltration in Tregs. While it may seem as though Tregs are largely responsible among the T cells to initiate pro-tumor responses, it is important to keep in mind that these properties of Tregs allow for them to be genetically re-engineered in order to use them for therapeutic purposes to fight the tumors, such as immunoediting. Thank you.